In this session, we're going to be going through Edexcel Combined Science Paper 2, Biology. This is from the year 2018, and it's incredibly important that you go through the whole paper until the end. If you're able to gain a couple of marks in every single question, by the end of the paper, you would have improved a whole grade. In question 1, we are given figure 1, and this shows a diagram of a red blood cell from a turtle and a red blood cell from a human. The cells are animal cells, and we've got to complete the sentence. Animal cells do not have A, a cytoplasm, B, a cell membrane, C, a cell wall, or D, mitochondria. And the correct answer would be that animal cells do not have a cell wall. In part 2, the actual length of the red blood cell from a turtle is 20.5 micrometers. Calculate the length of the magnified image of the red blood cell of the turtle when magnified 400 times. So to calculate the length of the magnified image, it is the actual length multiplied by magnification. And our answer works out at being 8,200 micrometers. In part 3, the width of the human red blood cell when magnified 400 times is 3.08 millimeters. Calculate the actual width of the cell and show your answer in standard form. So to work out the actual length, we're going to have the measured length divided by the magnification. And if we work that out, we get an answer of 0.0077. But the key here is we have to give our answer in standard form. So when we're looking at this, to give our answer in standard form, it would be 7.7 times 10 to the power of minus 3. Then for the next part of the question, part B, we're told that red blood cells are carried in veins and arteries. Figure 2 shows the equipment used to measure the elasticity of an artery. And we have our diagram here showing how the equipment is set up. Then part 1 says, describe a method you could use to see how much the ring of tissue from an artery could stretch before it no longer returned to its original size. So to do this investigation then, first of all, we are going to have to take the initial length of the artery. So we need to measure the length of the artery before we do anything. We would then add a 10-gram mass. We would remove the mass and measure the length again, and we would repeat this process until the tissue no longer returns to its original size. For the last part of question 1, we are asked to give one safety precaution you need to take when handling animal tissue such as blood vessels. So the obvious one is that you need to wash your hands, but you could also say you must wear gloves, or you need to sterilise the equipment after you've used it, or you need to disinfect the area after you've used it. A student compared the number of stomata on the upper and lower surface of a leaf. She completed a leaf peel as shown in figure 3. So figure 3 shows what was done. The leaf was coated with clear nail varnish. The layer of the nail varnish was removed, and then the layer of nail varnish placed on a microscope slide. So the first part of the question says the layer of nail varnish shows an impression of the cells of the surface of the leaf. Part 1, state why a cover slip is placed on top of the leaf peel, and the cover slip is placed to make sure that the leaf peel is thin and it's flat. Part 2, explain why the leaf peel rather than the whole leaf was viewed with a microscope, and we're looking at the leaf peel because it's much thinner, which means the light can pass through it and we will be able to identify the cells that were in the leaf. So for part B, the student drew a biological diagram of the leaf peel taken from the underside of the leaf. Figure 4 shows the diagram. State the number of stomata visible in figure 4. So we've got 1 here, 2 here, and 3 here. So the correct answer here would be 3. The student observed that the stomata were open. Describe how stomata open. So for stomata to open, the guard cells take in water by osmosis, and that causes the guard cells to be turgid, and therefore the stomata is open. The leaf peel from the upper surface of this leaf showed no stomata. Explain why it is an advantage to the plant to have this distribution of stomata in the upper and lower surface of the leaf. So the stomata is what allows gas exchange to happen, and gas exchange is what allows photosynthesis to happen. So the more gas exchange, the more photosynthesis. In question 3, figure 5 shows the estimated blood flow through some parts of the body when a person is at rest and during exercise. So we have the brain, the heart muscle, the digestive system, other muscles, and all other organs except the lungs. Part A is asking us to compare the rate of blood flow through the body when the person is at rest and during exercise. So we're not explaining, we're just comparing. So for this question, we would say that the blood flow through the muscles is increased during exercise. We could say that the blood flow through the digestive system decreases during exercise, and that the blood flow through the other organs stays the same. For part B, we've got to explain why there is a change in the rate of blood flow through the digestive system during exercise. So, because there is an increase in the blood flow to the muscles, because that's where energy is needed, the blood flow to the digestive system therefore becomes restricted. For part C, the stroke volume is the amount of blood leaving one chamber of the heart per beat. From which chamber of the heart does the volume of oxygenated blood flow? So our options are A, the left atrium. So that would not be correct because the left atrium receives blood from the pulmonary the pulmonary vein. B, that would be our correct answer. C, the right atrium, that's not correct because the right atrium receives blood from the vena cava. And D, that's not correct because D is where we receive deoxygenated blood. Then for part D, a person has a cardiac output of 4.9 litres per minute. The stroke volume of each heartbeat is 70 millilitres. Calculate the heart rate. So the first thing that we have to do is we have to get both of these into the same units. So if I'm going to go with millilitres, 4.9 litres as millilitres would be 4,900 millilitres per minute. And that would be divided by our 70 millilitres, which is the stroke volume, and that gives us an answer of 70 beats per minute. So for question 4, a student investigated the effect of nitrate ion concentrations on plant growth. She placed the barley seeds in 3 test tubes containing different concentrations of nitrate fertilizer. Test tube 1 contained distilled water with 1 pellet of nitrate fertilizer. Test tube 2 contained distilled water and 2 pellets of nitrate fertilizer, and test tube 3 contained distilled water and 3 pellets of nitrate fertilizer. After 7 days, the lengths of the seedlings were measured, and figure 6 shows an example of the apparatus used. So our first question is to describe a control for this investigation. So the control would be that we do this investigation in exactly the same way as it's shown in figure 6. So we put one of the seedlings on top of the cotton wool, but instead of having nitrate solution, we just have distilled water for it to be a control. The nitrate fertilizer contained the chemical compound potassium nitrate. The hazard symbol on the bag of potassium nitrate fertilizer is shown in figure 7. So what we need to do is we need to say which hazard does this symbol represent. So that is the symbol for an oxidizing agent, so the correct answer would be B. Part 3. Give a method other than measuring the length that would show the growth of the seedling. So if we're not measuring the length, the other thing that we could do is measure the mass of the seedling or the change in mass after 7 days. In part B, figure 8 shows the results of this investigation. So we have seedlings in test tubes 1, 2 and 3. We've got the lengths at the start, which were 4, 6 and 5, and then we've got the lengths after 7 days in millimetres, which were 11, 17 and 26. Part 1 says explain why there are differences in the change in lengths of the seedlings. So the answer here is going to be that nitrates are needed to produce proteins for growth. So where we have the largest amount of nitrates in the solution, we've got the largest amount of growth after 7 days. Now for part 2, we're asked to explain how nitrate ions were absorbed by the seedlings in test tube 3. So here the nitrates are going to be absorbed by the roots, and this happens by active transport. So the nitrate is going from an area of from an area of low concentration to an area of high concentration. Farmers use crop rotation to reduce the need to add nitrate fertilizers to the soil. Plants such as peas and beans have a mutualistic relationship with nitrogen-fixing bacteria. Explain why farmers use these plants in their crop rotation. So the peas and the beans, they increase the amount of nitrate in the soil because they have nitrogen-fixing bacteria which live in their root nodules. So the pea and bean plants will then naturally produce nitrogen compounds because they have nitrogen-fixing bacteria living in their root nodules. Figure 9 shows the effect of light intensity and temperature on the rate of photosynthesis. So part 1 says to describe the effect of light intensity on the rate of photosynthesis. So here we can say that as the light intensity increases, the rate of photosynthesis increases until light intensity has stopped being the limiting factor, and that's where the graph levels off. So for part 2, we're asked to explain the effect of the temperature on the rate of photosynthesis. So here, as the temperature increases, so does the rate of photosynthesis, and that's up to the maximum or the optimum temperature at which the enzyme is able to work. After the optimum temperature, the enzyme will become denatured, so photosynthesis decreases. Now, for this question, we are told that a student measured the rate of photosynthesis using algal balls in the laboratory. The tube of the algal balls was kept at temperature of 25 degrees Celsius and was moved to different distances from the light source. The result of this investigation showed the rate of photosynthesis is, and we've got to choose from either A, B, C, and D. So A is directly proportional to the distance of the light source. B inversely proportional to the light intensity, C directly proportional to the temperature, or D inversely proportional to the distance from a light source. And the correct answer here would be D. It's inversely proportional to the distance from the light source. So for part C, we need to explain how substances are moved through a plant by transpiration or translocation. So if we look at transpiration first, transpiration is caused by the evaporation of water from a plant surface, and transpiration happens mostly at the leaves through the stomata. Now, the loss of water will create a shortage, so water is drawn up through the roots and then through the xylem, and the water flows in one direction to where it is needed. Now, when we think about translocation, translocation is about the movement of sugars from the leaves where they're made by photosynthesis. And translocation will happen throughout the plant as the sucrose is moving through the plant. Now, this will happen through the phloem, but the phloem at the ends will have pores at the ends of the cells, and this will only allow certain things to go through the pores. The phloem, however, will allow direction of the sucrose in both directions, so the sucrose will end up where it is needed most in the plant. Figure 10 shows the concentration of hormones estrogen and progesterone in the bloodstream of women at different ages. So use the information from Figure 10 to explain why women over the age of 50 are less likely to ovulate. So if we look at the area of round about 50, we can see that the levels of estrogen are extremely low. And if the levels of estrogen are extremely low, there would not be a message going to the pituitary gland to release the luteinizing hormone. And it's the luteinizing hormone, the LH, that's needed for ovulation of the egg to occur. Now for part 2, it says use the information from figure 10 to explain why women are less likely to menstruate after the age of 60. Okay, so when we're looking at the age of 60, we can see again, because the estrogen levels are extremely low, that means that the lining of the uterus would not build up, so there's no lining to be lost in menstruation. Explain how clomiphene therapy may increase the chance of a woman over the age of 50 to become pregnant. So during this therapy, what happens is the clomiphene therapy causes the release of the luteinizing hormone, the LH, which then stimulates the ovulation of the egg. The hormone progesterone is produced by so the gland that produces the progesterone is the corpus luteum. And then explain how the release of adrenaline can result in the improved performance of an athlete. So, adrenaline will increase your heart rate. And if your heart rate is increased, that means there's more blood flowing around your body. If there's more blood flowing around your body, that means there's more oxygen and glucose going around your body. And if more oxygen and glucose is going around your body, you are going to respire more, and respiration will therefore release more energy. And that brings us to the end of this past paper. I hope you found this useful. If you did, please comment below that you did find this useful and you'd like to see more of these past papers being uploaded. Also, if you'd like to subscribe, what that would mean is that you would have easy access to all the new papers that are being uploaded on a frequent basis, as well as more learning material for science. I look forward to seeing you in some more of these videos, and I wish you all the very best in your learning and assessments that you may have coming up.